Good morning, everyone. I'm Randy Hubbell, CEO for Carmell Therapeutics. Carmell is a regenerative medicine biotech / medical device company. We are creating value by taking multiple assets through the clinical regulatory pathway for BLA approval through CBER. I spent 13 years at J&J. The last 5.5 was with J&J Biosurgery, where we took plasma-based materials and created medical devices such as fibrin sealant, fibrin pad, Evercell, and Everest. A lot of those were through the BLA process. Dr. Jim Hart retired from J&J as the Chief Medical Officer for J&J Global Surgery. He's our Chief Medical Officer. Donna Godwood retired from J&J as Chief Quality Officer for J&J's entire medical device and diagnostic business, a $30 billion business. She's our Chief Quality Officer. Dr. Stephanie Kladakis is our Chief Scientific Officer. She's a Harvard undergrad, PhD in tissue engineering, taken many technologies from concept through development. And Dr. Vargo also retired from J&J, and she runs our clinical trials. So here's the update on the company. Because we are a BLA, we go through the IND process, investigational new drug. We did get IND clearance to begin a pivotal study in our lead program, which is a bone healing accelerant for accelerating the healing for tibia fractures after very, very severe injury. We are a platform technology. We have, as you'll see from our pipeline slide, multiple assets that are going through the process. Our core technology all works very much the same in it that it allows for the proliferation, enhanced proliferation of cells to regrow tissue, either bone, soft tissue, skin, hair, collagen. So it's very much a platform technology. We're regulatory and clinical centric, meaning we're doing the hard work of creating the regulatory approvals and clinical trials that is necessary for the world that we live in today to provide the necessary evidence in the robust clinical studies. We have an experienced management team. Our manufacturing is completed. That's a big part of a BLA, is your CMC and getting that organized before you start your pivotal studies. So the technology is placed locally at the injured site. We will have our first product you see up there in the middle is a putty consistency. The second product is more of a paste. We have demonstrated that we can accelerate healing, enhance healing, and reduce infections in both humans and in animal studies. So what is the technology? It is allogeneic plasma, platelet-enriched plasma. So the market's very familiar with autologous PRP. This comes from blood collection centers. The significance of the technology from Carmell is we discovered a way to process it for safety, viral inactivation, without destroying the proteins, without destroying the regenerative factors, without destroying the growth factors. We keep the plasma whole, so all of the regenerative factors are there. Tremendous amount of science has already been shown for growth factors, PDGF, VEGF, IGF, EGF, all of those growth factors are present and we count them and we know they're present. That has to be done as far as the BLA. So we, significant discovery there. We also have the ability to plasticize it. What that means is we take fresh frozen plasma, we pool them together and then we lyophilize it, it becomes a powder. That powder is our drug substance. In the drug world, that would be very similar to API, active pharmaceutical ingredient. The cross-linking, big deal. The cross-linking is a material that was discovered by the scientists. We cross-link it with genipen, a derivative of the gardenia plant. It provides for presence. All other forms of trying to deliver growth factors right to injured sites have washed away within hours or a day. This will be present and degrade over time. That cross-linking protects the proteins from the body's process of degrading it. So the first product will degrade over about 8 weeks. We have consistent bioactivity. We'll deliver the same level of bioactivity on each lot. It's off the shelf, ready to use. Here's our pipeline. We'll start with long bone fracture. We'll be moving into a pivotal study this summer and stagger the second pivotal studies. With the BLA, you need to have 2 studies demonstrating superiority over standard of care to get an approval. We also are pursuing a CE mark in Europe. We are a class 3 medical device because our first product has beta tricalcium phosphate in it, which is specific to bone. And we'll start that process by submitting under the new MDR this year. And Have no idea how long that's going to take. As some of you may be familiar with the new regulations, it's a lot of unknowns, but we'll get the process started. Good news for us is we have data. We have phase 2 data that we can submit with, and we'll have a bunch more data coming after that. The indication will be a bone void filler in Europe. We'll pursue the spine indication, dental. An interesting one that has really been accelerating over the last couple of years is for androgenetic alopecia. That is male pattern baldness or hair regrowth. That is a procedure that's being done today. They do microneedling with radiofrequency microneedling, creating pores in the scalp. They do the same thing for facial rejuvenation. They take the patient's blood, spin it down, and take the top coat plasma enriched with platelets and push it into those pores. Our product will be a substitute product. It will be off the shelf, ready to use, no blood draw, and we'll have the benefit of a guaranteed bioactivity, and we'll have the benefit of a slow released mechanism that doesn't exist today. So we have hope that it will perform even better than the results they're getting today in the aesthetics market for hair regrowth and facial rejuvenation. So here's our evidence. We have both human and a number of animal studies. The 8 core animal studies that were conducted were for the purpose of regrowing tissue, accelerating healing, reducing infection, regrowing bone. All of that supports our phase 2 data that the FDA has used to allow us to go into a pivotal study where we studied long bone fractures. So these are patients that come in with open tibia fractures that need to be fixated with hardware. We don't replace the hardware. We're an adjunctive too. Our product gets pushed into the cracks and crevices of the bone. That study showed that we were able to accelerate the healing, reduce infections, and also we were able to show a tremendous improvement in the wound healing. So the way the study was designed was 30 patients, 20 treatment, 10 control. They presented with tibia fractures coming through the skin. The patients were then fixed with hardware, either internal or external hardware. Then they were randomized. If they were randomized to treatment, the product was taken off the shelf, ready to use, pressed into the cracks and crevices of the bone. The patient was closed, and then they monitored them at 3, 6, and 12 months with an X-ray, an AP, and lateral view. And when you do that, you get 4 views of the cortices, and they looked for the healing of the cortice of 75% or greater bridging. And when you look at that, you see that circle there. We did, in a very small study, hit statistical significance of a greater mean number of cortices that met that definition of healing radiographically in the treatment group, which is BHA, bone healing accelerant, versus the control. The other analysis that we did was a post hoc analysis, had to reread according to a new scale, an upgraded scale, radiology scale called RUST, Radiology Union Scale for Tibia Fractures, in which they look at each cortice on a scale of 1 to 4. 1 not healed, 4 healed. So each time point, the minimum you can have, scores you can have is a 14, the maximum is a 16. 13 is considered healed. When you combine that with patients that were ambulatory and had no secondary intervention for a composite endpoint, all 3 need to be true. None of the control patients met that definition, and 36% of the treatment group met that definition in the most severe 3A and 3B patients, which is a severity score. That is our that is our pivotal study design. We will be using the RUST composite score, and we expect to see hopefully a 36% or greater treatment effect. We also saw a tremendous reduction in infection rates as the soft tissue sits right on top of the bone and was bathed in those regenerative factors. Most of those infections were not deep infections, they were superficial, so severity was low, frequency was high in the control group, but the frequency was not in the treatment group. That was supported by the animal study we did, where we took a rabbit model and infected a pacemaker pocket with MRSA, and we saw that our technology by itself was able to have all the rabbits purulent free and half of them culture negative for MRSA. So the technology has a couple of different indicators that it could be a tremendous impact on reducing infections. So we will be sold in the marketplace like a medical device. These will be surgeons and wound care folks. But our program is very much looks like a biotech company. So we'll be advancing a number of our areas through different therapeutic areas: bone orthopedics, dental, advanced wound care, aesthetics. And with that, it creates an opportunity for us to create partnerships. So that's what we'll be doing over the next 18, 24 months, is looking for the right strategic partners that have global capacity to distribute the technology. Fortunately, our cost of goods are very low, allowing us to have both parties, the OEM, us, and the strategic distributor, both enjoy great margins. Thank you.